could start a range of projects related to the study of genomic history of Armenians. You have heard excellent talk yesterday from Levon, and also direction of studying the distribution of genetic variants linked to various diseases in Armenian human genetics, which was started by our laboratory of human genetics, genomics and immunomics. So our efforts, of course, with the great help of Arsen Arakelyan, maybe he's a general partner of New Venture Capital Venture Fund, and also he was involved at the very early Human Genome Project back in 30 years ago. We finally somehow managed to transfer the project to the government, and we had a discussion on December 2018, and during this discussion on governmental meeting, project was approved, and it is about to launch. Hopefully it will launch at the end of this year. There is plenty work to do, but at least we hope that foundation, Armenian Genome Project Foundation, will be announced, and we will have governmental decrement. So what is the main goals of Armenian Genome Project? Actually, there is threefold project. First, actually carry out Armenian Genome Project, which is population level sequencing for Armenians, in order to get Armenian reference genome. Because as you have heard yesterday, we have there is significant variations between different populations in terms of either genetic markers that shows the ancestry or genetic markers that lead to disease development or predisposition. The second, after completing this part, we will proceed to the Armenian Cancer Genome Project. Basically, it's not just cancer; it's any chronic disease which has interaction of genes and environment, in order to understand what is the distribution of these risk alleles for genes that are linked to diseases which are prevalent in Armenian population. And to do these two things, we need the third one, which is the most important. We have to have here sequencing facility. I'm pretty sure that you know that there are several sequencing machines in Armenia, but all of those are small-scale machines that are not capable to perform large-scale whole genome sequencing. And the idea is to establish here an infrastructure which would be very unique for the region currently, and use this infrastructure to implement the project. So if we go to the tasks, the first task will be drafting Armenian genome. So basically, what is Armenian genome? So we know that we all belong to Homo sapiens species, so we basically share the same genetic information, but Armenian genome refers to a set of genetic variations which are more prevalent in Armenians compared to other populations. And a number of national genome projects which are already launched were succeeded to show that these variations and change in preference presenting in various populations. So we will characterize genetic variations among Armenians. We hope that this study also will contribute to more deep understanding of genetic continuity in this region and more deep tracing of Armenian genetic history. Next step would be assessment of population-specific disease risks in terms of what are the most prevalent risk alleles in Armenian population. We'll try to identify some population-specific markers for disease development that will be then somehow used not by the project immediately, but by creating the resource for others to use this information to develop prognostic and diagnostic tests. And also we'll use this information to understand mechanisms of disease development. So basically the best brilliant example of this is Iceland Genome Project implemented by dCODE, which identified several markers for Alzheimer's disease, and now these markers are part of clinical diagnostics for the late or early onset of Alzheimer's disease. it's not just science we are pursuing in this project. We believe that this project will have much more wider impact in Armenia rather than publishing several papers in very high impact journals. So important thing is R&D infrastructure. So we believe that building Armenian Genome Sequencing Center and implementing Armenian Genome Project will see biotech and pharma industry here. So it will be just an initial step to attract pharma companies here, to attract biotech companies here, to create an atmosphere where biotech and genomic startups could develop here. From scientific point of view, I think everything is very obvious. We will get a lot of data, we will analyze it, we will get a lot of results. This result will be highly publishable, published in very good journals, and this will promote like ranking of Armenia, scientific ranking of Armenia. From societal and healthcare point of view, even yesterday with this limited amount of data Maria demonstrated you, we can see that we can able to prognose incidence of this or that disease based on genetic risk profile, like population specific genetic, it's called population risk scores. And this could be used to tune up the public health program. So if we know that there is a much more risk of having diabetes than, for example, schizophrenia, the government should implement programs related to prophylaxis, like more resources should allocate to prophylaxis of diabetes rather than addressing something with schizophrenia. Economic impact is really linked to R&D infrastructure. I already told about this, so we expect that it will really boost genomics, genomic biotech in Armenia. From educational point of view, we will be able to have a structure which will constantly provide trainings and education to all students who are involved in the field of biomedicine. So there are several programs like Russian Armenian University Bioengineering and Bioinformatics, at the American University Data Science with Bioinformatics track, at the State University Whole Biological Faculty, Medical University, for example, Clinical Genomics. So all these students could be targeted within this program. They can learn and become expert in the field of genomics. And also what is last but not the least important is that currently we don't have any legislation about privacy and protection of genomic information in Armenia. We don't have any legislation about discrimination based on genetic background. And this project will help to frame these regulations. So we will achieve the level of protection of genomic privacy that now is present in the US or in Europe. So basically the project is continuous, but if we just divide on phases, we will have first phase with 3,000 genomes sequenced. Next phase we will proceed to exomes, genomes, and also clinical exomes, so exomes for diseased people. And it will ongoing, and we intend to collect as much data as possible. What technology lies behind this genome project? The technology is called next generation sequencing. So many of you know that there was the first generation of sequencing, which is called Sanger sequencing. Next generation allows massively, in parallel way, process DNA and get entirely much more output of data than you can do it with Sanger sequencing. So basically you take sample DNA, fragment it into small pieces, then you sequence these pieces with various different techniques depending on the technology provider, and you get something which are called short reads. So these are short information or sequences for short stretches of DNA coming from this original DNA, and then the workflow is to align these reads onto reference genome and to see whether there are some variations within this. So whether alignment is good, so if you can able to cover whatever specific region of human genome, and to see if there are any variations within this in this particular sample compared to reference genome. So technology started again, so it was very costly at the very beginning, but now prices are dropped like dramatically. Currently you can have whole genome sequence for $800 US. Yesterday at the airport, when I was meeting one of our colleagues waiting there, I found an article that one US company dropped prices to $500 US. So you can have your entire genome done by $500. And if we compare with Human Genome Project, it took 10 years and $3 billion to build up one genome. And now you can have yours for very reasonable price. And this opens a lot of opportunities. We can study what kind of genetic predispositions do you have, not just like any human, but exactly you. What you should be aware of, what you should know, how you should react in case of this or that disease. But this, again, comes for some cost. And this cost is the resources needed for data analysis. So imagine each time you generate a huge amount of data, and this huge amount of data should be somehow analyzed. So you can't read entire your genome. You should concentrate on specific parts which are informative and can tell you anything about your, something about your biology or phenotype. When I say it's resource hungry, this is what I mean. So if we start the whole workflow, so the first file you will generate will be about 100 gigabytes, then the next file will be about 300 gigabytes, and basically for one individual, for one person, you will get 1 terabyte of data. And if we have 3,000 genomes, so it will sum up to almost 2 petabytes of data. And this is a huge amount. It also, of course, collects the raw files. You shouldn't throw them out. You should keep them because then you want to trace back. It would collect, also store some intermediary files. But anyway, so you would need this much place to store this whole data. And you need not only store this data, but also analyze. Suppose you have a very good computer, but it's desktop computer. So with 16 cores, 32 gigabytes of RAM, basically Siddharth's dream of having this. But even with this computer, you will spend 48 hours to analyze one genome. And with 3,000 genomes, you should be very, very healthy to see the results. This is the example from Dutch Genome Project. It's called Genome of Netherlands or Dutch Genome. So they analyzed about 750 samples in trios, so in nuclear families, mother, father, and child. So data was stored in 500 terabytes. Overall storage was 2 petabytes. So the actual data, their backup and intermediary files. They use a cluster of 10 Linux machines with 48 cores and 256 gigabytes of operating memory. It was heavily parallelized. And even with this parallelization, it took about a year to analyze this data. So this is what we will face. And I hope that we will have this opportunity since we'll get soon, I hope that soon we will get a large and very powerful supercomputer which was donated by the University of Toulouse. This is from bioinformatics perspective. This is the workflow. So we'll start with raw files, we'll do several different steps, and then these are all boring steps. They are very needed, but they are kind of boring. Interesting things are here, and you have heard a lot about interesting things yesterday. If we zoom out from the workflow, we can actually think about alignment, calling genetic variants, their annotation, and high-level analysis. So what is alignment? Again, you have a lot of stretches of short reads. They look like this. They could be longer, they could be shorter, but basically about 100 or 200 base pairs. And you have to somehow arrange them on the reference genome in a way that it's very similar, so you can have each read aligned as perfectly as possible to the reference genome. The reference genome is the one that was generated during Human Genome Project. And this allows you to get this very perfect arrangement, but if you always, at every step, you try to perform quality checks, because otherwise you will miss something. And then at the end you will get some unpredictable, unknown results. And you should be very careful about insertion and deletions, because single point variations can be handled very easily, but these small insertions and deletions, which we have actually a lot in, or all of us have a lot in our genomes, they could be dangerous, they could be very neutral, but they are there, and they can cause significant variations in alignment. After you get your alignment, then you simply trace whether nucleotides in your short reads correspond to nucleotides at the reference genome. And whenever you see a difference, for example, in this case, in all short reads we have here nucleotide G, on a reference genome we have A, this could mean that there is genetic variation. And this is mutation that occurred in this particular individual. And this is actually what we are looking for. So this is just one small part of genome, and if we go, like, extend our analysis to a level of gene, we'll get this kind of picture. These all small gray and blue and red things are short reads, and the bars indicate mutations occurred at any position within the gene. And we record this information. And it's very why we need national genome projects, because we want to each time to identify novel mutations. So how it happens? Either these mutations were never seen in other population and it's not reported, or it has very low frequency and it's also not reported. So each national genome project actually ends up with a bunch of novel genetic variations. And these genetic variations could be either, again, have some impact on phenotype. For example, it could be aging, food preference, or disease susceptibility, or they will be very neutral. Basically, we all carry a lot of mutations inside, and which are not visible. Some of them are visible, they constitute the phenotype, but the rest is simply there. Singletons are also a very interesting point. It's mutations that happen within single individuals. So on the population level, their frequency is very low, but you can find them in different persons. And also, interesting point to control is accessible genome. So not all parts of our genome could be sequenced in the same quality. Some parts you will get much more coverage, much better sequence, much more precise sequence. Some parts you will not. And you should control to understand what part or what percentage of your genome was actually sequenced and what did you miss. For example, you can miss a whole bunch of your telomeres, which are very important information. It's directly related to aging. But thanks to Lilith and our group, we developed software that can actually measure it. What are issues? So each step has its own specific issues, and there are also common issues. So what are the specific issues when you are doing variant calling, or trying to identify mutations or variations in your genome? First, you should always validate your program you use for identifying these mutations with some highly confident reference set. So there is a reference set which were analyzed using gold standard methods, and they are available. And you should be able to reproduce the genetic variations reported within this set using your alternative bioinformatics pipeline you have developed. You should always use not one but several pipelines. Each pipeline has its own advantages and disadvantages, and more pipelines you use, more you can say for sure that variation you found is real variation, but not some mistake in your analysis. Whenever possible, you should do also comparison with other genotypic methods. For example, we plan in Armenian Genome Project, among samples we will collect, also use samples which were analyzed in different places, and we have their genotype information, just to check whether we can do good laboratory work. Always you should use the newest version for the software, because this all software, the bioinformatics software, mostly is open source, so there could be a lot of bugs, whatever software you use, the first it comes disclaimer that there is no guarantee that what you get is what you really want to get and it's correct. So, but they are getting improved. They are improving. So whenever you use the newest software, the more chances that what you see is really what you see. And if you do population level discovery of genetic variations, the chances that you get positive detections are increasing. When you work on single people, like individual, chances are lower. You get this is what you get at the end. So this is raw, so-called variant calling file. So you have your chromosomal number of chromosome, position at the chromosome, what you should expect, and what you get there. So it says nothing, yes? Basically, it's just a position and a letter substitution. So what you should do, you should annotate this everything. And annotate using a variety of databases. And finally, during the annotation process, you get this. So you get its ID if it's there, what gene it belongs, what is the consequence of this mutation, is there a disease which is linked to this mutation, and what is the effect of mutation. Is it pathogenic, neutral, or whatever. And here the problem is mapping like this. There are like plenty of databases now in genomics world. And the same thing there could be annotated in like 1,000 ways. And you have to have some clues how to map one annotation to another. The same SNP could be reported in 10 names. And you have to be able to collect them into one place. So this is very much data science and issue to create a universal identifier that will help you to find out gene or whatever markers you are looking for. So, and then if everything is good, you go to high level analysis done by Levon and Maria. You have heard these talks yesterday. And with this, at least the part which I'm also very much interested in, there is a growing, so we're at the very front of this direction. So there are only few papers appeared that try to utilize population-wide genomic information to predict healthcare issues. And so we are in a very good position at the moment. But besides the specific issues, there are also common issues. And common issues are storage, so we have to have plenty of storage. Computational speed, we need to be able to compute data very fast, because otherwise we will wait for decades. Privacy and protection, so your genomic information is your private information. Even if you agree to share it, reasonable effort should be paid to protect your identity. And this is like something of topic of no discussion. And also visualization, so you get your data, but you have to somehow see and comprehend it. So storage issues, let me skip. What is good with computing is that a lot of tasks could be parallelized. So there is in parallel you can align short sequences because you have a lot of them and they are rather say independent from each other. You can search in databases, so you can search one string in a lot of databases and you can do this also in parallel. Statistical tests and a lot of other things that could be done in parallel. And now more and more software, they switch from the single processor using mode to parallel, to using clusters or parallel threads or things like that, and also using graphical computing units like CUDA, NVIDIA graphical processors, or cloud computing. More important issue is privacy. So again, it's your data. And using this data, actually using this data is very important for science and healthcare. So we should get data to be able to analyze it and then come back to the people and say, okay, these are the very important markers you have to diagnose in this disease or that disease, or these markers somehow are related to disease outcome or selection for therapy. And so we need this information. And it's good that people are willing to give donate their genetic information. But this data should be protected. And currently, it was shown that having your DNA profile, you can be identified up to surname. So also, it's not only your data, it's also your offspring data. So basically, your offspring should also somehow agree that you provide their private information. It can go to absurd, but this issue is there. And there is a lot of regulations in the USA and in Europe about these topics, but only regulations are not enough. It should be, regulations should be accompanied by some computational tools that will allow to keep this privacy. For example, this is a paper from 2013. So having genomic information about personal genomes, some open databases, you can trace person up to its surname, up to its address and a street. But this shouldn't be done. And you can, but you shouldn't. And because not all people understand that they shouldn't, there should be also mechanisms for preventing this. And for example, now it's a start for developing secure or privacy preserving computational tools. For example, this is a tool or framework for computing genome-wide association data. So all data is encrypted. There is no way to decrypt it, but still there are algorithms that can analyze this encrypted data and produce results. And these results cannot be traced back to people, to the persons or samples that were used within analysis. Similar thing is secure privacy-preserved medical testing. So you want to have a medical test, but you don't want to expose any additional sensitive data. And there is a way of doing this. So again, new frameworks are being developed that basically will allow interaction between patient and doctor, but will protect other people to accessing this data in a good way. Yes, but still, this data could be used by researchers. Even on the level of alignment, you still can expose your data when you simply try to align your data to genome. So another area is to start protection from very beginning and design secure aligners. So this would again align your short read to the reference genome, but in a way that it could not be exposed to other parties. Visualization. So basically, all people like good pictures. Biologists very much admire pictures that looks nice but tells nothing, because you will not probably be able to see here anything. And these are actually term-point reticulogram, and it's used a lot. But basically, that they don't bear any functional information. So here, whatever you propose, which would make sense of having nice information, would be appreciated. For example, we have heard a lot about very good visualization methods offered by our colleagues from Leipzig University, and I can read and understand this data. For me, it's good enough visualization. So if we summarize the key technological needs, so if there are people coming from machine learning, data science, so please consider these topics. We are very interested in good ways of data acquisition, good and secure ways. And it's not just genomic, but also phenotypic data, imaging, real-time data. We need as much data we have, as more precisely we can then link genotype to phenotype. We need new technologies for data storage, much more efficient, because now it's petabytes. It will go to exabytes, it will go to whatever then later is coming. We need good technologies for computing, which will be memory efficient, minimize data movement. Basically, data movement also very big issue. You can get this 2 petabytes data, but if you have to transfer it over internet somewhere else, you will surely not see even the upload progress again. So we need fast ways to upload data, fast ways to analyze it, and memory efficient ways. We can't afford having 1 petabyte of operating memory now. and anything else that could help. And literally anything else that could help, and it's good that genomics, data science, and technology now are emerging, and I hope that these all efforts will really help at some time to get much better tools to fight diseases and aging than we have at the moment. So with this, I would like to thank you very much for being here with us, and specifically thanks to our collaborators and members of my lab. You can find more information about Armenian Genome here at links, and of course, agencies that funded this conference and all. Thank you.